Might work, I think. Okay. Recording. There. I think we're good. "15 seconds to spare." Thank you. Do this. On. Okay. There. Now I think it's coming around. Good grief. There's still some people waiting. Okay. Let's let them come in. I'm going to go ahead and try to get started anyway. Let's see. You guys, this is you right here. And let me fix my display here. That looks good. All right, let me catch the lights. Finally. That looks good. All right. Finally, we're getting started. Good. So today is Tuesday, February 2nd. We're 2 days into February. I think today's Groundhog's Day. Did y'all hear that? So Pete saw a shadow, so 6 more weeks of winter, I guess. So is that a good thing or a bad thing? No, everybody wants winter to be over with, so I guess that's not a good thing. He sees his shadow and I guess it frightens him, so he goes back in his hole and he hides for six weeks. Anyway, but you guys are Biology 1160, Section 2. This is my Tuesday, Thursday class. Like I said, this is Tuesday, February 2nd, 2021. And so let's get started. It's, oh gosh, 3 minutes after 11 already. So last time, I think we stopped right here. I was gonna do the cartilage, but then said, nah, I'm not gonna do it. So I'm gonna do it all at one time with a fresh start. So right here, remember, we're talking about connective tissue. There's epithelial tissue, connective tissue, muscle tissue, nervous tissue. And this is connective tissue. And then had like 4 categories of connective tissue proper, then the cartilage, then bone, then blood. So cartilage is in its own group. And as you can see, there's 3 different forms of cartilage. What makes cartilage cartilage? So across the board, there's a couple of characteristics that all cartilages have. And if you understand this first one, hyaline cartilage, the other 2 will be pretty easy, because the other 2 are basically just modified forms of hyaline cartilage, okay? What hyaline cartilage has, it's like the basic unmodified or most primitive form of cartilage is it has a solid matrix. Remember, all connective tissues have cells, proteins, and a matrix, okay? Well, its matrix is solid. It's more like, I always compare it to, I'm still admitting people, okay? Almost like solid rubber, like a rubber bumper. And rubber is solid, but it's not hard like bone or rock or stone or something like that. It's somewhat pliable, and that's typical of all cartilages. Okay, now it's a tissue, and so like tissues, it has to have cells. But since it has this solid, firm, kind of rubbery matrix, the cells have to be accommodated with their own little environment. And so if you look carefully in the matrix of all cartilages, you can, it shows really well on hyaline cartilage. You can see that it's pitted. You see this photomicrograph right here? It almost looks like blue Swiss cheese or something. And those little bitty holes you see, little pits, are called lacunae. Or singular would be a lacuna, L-A-C-U-N-A, which means little lake, like a shad, little pond. And what these lacunae are little bitty, little pits or little cavities of interstitial fluid to accommodate the cartilage cell. So inside each one of these little lacunae is a cartilage cell or a chondrocyte. And so all cartilages have chondrocytes in lacunae. Now the difference is, you'll have proteins out in the matrix or not, okay? This first form, hyaline cartilage, means, hyaline means glass. So it's clear, it's almost translucent, where light can pass through it pretty easily. But the other 2, it's more occluded. And so the other 2 forms are actually possess one of the 2 types of protein fibers. Remember, we had 2 protein fibers. We had elastic fibers and collagen fibers. Well, hyaline cartilage lacks fibers. Elastic cartilage has elastic fibers, and fibrocartilage has collagen fibers in the matrix. And that's the difference between the 3, really. And so, what do they look like? Where do you find them? What's their purpose? Well, the first one, hyaline cartilage. Remember, it says no protein fibers in the matrix. It's just chondrocytes and lacunae, and it's pretty clearish looking. And the example they show you, it's kind of a weird. They show you like a little fetal skeleton, which is true. Most of your fetal skeleton is cartilage before it's bone. Okay, and so it actually serves as a bony template. So when you're born, like I said, most of your long bones and other bones are hyaline cartilage, but over time they ossify or they turn to bone. Okay, and so that's a good location for that, you know, the fetal skeleton, I guess. But what about the adult? Well, you retain hyaline cartilage in certain areas. For example, you know, most of your skeleton as an adult is bone, okay? Osseous connective tissue, as we call it. But parts of it are still hyaline cartilage. Like, for example, you're in your nose, you got bony portions of your nose, but then the cartilaginous portion of your nose. You're in your ribs are half bone, half cartilage. So the cartilaginous portion of your ribs are called costal cartilages. And that's, remember, costal means rib. So the portions of the ribs that actually articulate with the sternum are hyaline cartilage. And then what we call articular cartilage. The word articular just means, forms a joint or an articulation. So in most of your joints, where one bone articulates with another, where they meet bone to bone, well, there's typically hyaline cartilage separating that. And so it cushions, remember, it's kind of like a rubber bumper. It cushions the ends of those long bones where they articulate. So you have a nice smooth cushioned surface, okay? And then in some organs, you'll see remnants of hyaline cartilage, particularly like in the histology lab, if you're looking at tissues under the microscope. For example, your voice box or your larynx, your larynx is made, it's all cartilaginous, but it's made out of 9 separate cartilages that are all named, and we'll talk about those later. But 8 of those 9 are hyaline cartilage. The 9th one, well, what is that made out of? Well, I'm going to tell you in a minute, but it's a different cartilage. But most of the cartilages in the voice box are hyaline cartilage. And then basically the inferior extension of the voice box, your trachea, is equipped with 16 tracheal rings. And so these little rings that support the trachea are hyaline cartilage. And then if you follow the trachea out into the lungs, they branch into bronchi. And supporting the bronchi, there's also little bronchial rings or hyaline cartilage in there supporting those as well. So where else? I'm trying to think of some other places that you would find hyaline cartilage. It's by far the most abundant cartilage in your body. And like I said, it's the primitive form in the sense that it's, kind of what you start with, the ancestral type, and then it's been modified or specialized in certain areas. How's it modified? Well, with one of the 2 different types of protein fibers, okay? The second one, it says elastic cartilage, okay? Do not confuse elastic cartilage with elastic connective tissue. Those are 2 completely different tissues. Now, they are both connective tissues, but elastic cartilage is a type of cartilage. All cartilages are connective tissue, but elastic cartilage is a type of connective tissue, a cartilaginous connective tissue that incorporates elastic fibers, okay, just like elastic connective tissue does. Remember, that's the stuff that's in blood vessels and in the lungs and so on and so forth. Well, what's this? Well, think about hyaline cartilage, where it's found and what it does. It's basically a skeletal element portion. It's part of the skeleton supporting the bones or substituting for bone or whatever. So it's a internal structural element. Well, here in elastic cartilage, it's the same thing, internal structural element, but you're marrying those characteristics with the characteristics of elastic fibers. Remember, stretch and recoil. So it can bend. So this is a pliable type of cartilage. It can change kind of shape. Remember the slinky? It can stretch out and recoil. Well, there's only 2 places in your body where you need this skeletal element to be able to kind of bend and snap back, okay? And that's in your ears, your external ear, and then the epiglottis. Remember the 9 laryngeal cartilages I was telling you about, the cartilages in the larynx? Well, one of those cartilages is called the epiglottic cartilage. The epiglottic cartilage is what's in the epiglottis, and the epiglottis is that flap that closes over the opening into the larynx when you swallow. The opening itself is called the glottis. So get it, epiglottis is above or over the glottis. So the epiglottic cartilage, it's made out of elastic cartilage because it's pliable. You know what it reminds me of? Like a Tupperware lid, like a Tupperware plastic bowl with a lid that snaps on there and it makes it watertight, seals it real good. And so that's what the epiglottic cartilage does. When you swallow, it makes a nice tight waterproof seal over the glottis to keep it from leaking. And that's the only place you have it. That and then your ear, your external ear. You think about these things on the side of your head, you can bend them, twist them, they'll change shape, but as soon as you let go, they snap back to the original shape. And that's a pretty handy dandy tissue to have for these things that are attached to the side of your head. Now, human ears aren't really that big. Some people have some big old ears, but think like ears like on a deer or some kind of other animal that's running through the woods, and it can bend down on a branch or whatever, but it'll snap right back into shape. So it's, get a protective type of thing. So, but that's it. The elastic cartilage, only two places in the human body, the epiglottis or in the epiglottic cartilage. And then the auricle, the ear. It's called auricular cartilage. Of course, the word auricular just means of the ear. All right, but the word auricular isn't a type of cartilage. It just means of the ear. So anything could be auricular. Okay, and then the third type, again, a modified form of hyaline, which now you incorporate the other type of protein fiber. When the other type, you only have two, elastic fibers and collagen fibers. So this is the collagen fibers in the matrix of the cartilage. And so you think of the characteristics of collagen. Remember, elastic is the springy stuff. But collagen is like the analogy always gives like a toe strap, really heavy duty, something you can pull on and it's stress resistant. It's pliable, you can bend it, but it's really, really, it's not springy. You can bend it, but it's really tough, okay? And so you think about this. Why would you have to have these tough, heavy duty, really fibrous structures in the matrix? Well, the analogy I always give for fibrocartilage. Now, look at this spelling, fibrocartilage, not fiber cartilage. Don't say that. Don't say fibrocartilage. It's fibrocartilage, one word, by the way. And that's what has collagen fibers, okay? And the analogy I give for this is that it reminds me of rebar. You know what rebar is? If you ever seen somebody build a house, they have a dirt pad where they're gonna put the house, but on the dirt pad, they build a foundation to pour the concrete. So the house is gonna sit on a slab. And so before they pour the concrete, they build a box to hold the concrete. And then they put all these metal rods in there. It's rebar, reinforcing bars, I guess that stands for. But why do they put rebar in there before they pour the concrete? You don't use rebar every time you pour concrete. Like a sidewalk, there's no rebar. Parking lots, they could be in the highways where you got heavy trucks going. The idea is that the rebar, the concrete in it, it helps reinforce the concrete, because concrete, it dries and hardens, it gets like stone, but it takes years, believe it or not. You think, you can start building on your slab in 14 days, or you can start driving on it in a couple of weeks or whatever. It's not really dry. Concrete takes years to dry. Sometimes like a concrete bridge, like 30, 40 years before it's fully dry. But as it dries, it condenses, shrinks, and it's going to crack. You talk to any concrete contractor, they'll tell you, yeah, it's going to crack. Concrete always cracks. Well, if you got a big old heavy house sitting on concrete, it's going to crack, but you want to minimize and direct the fractures, with these reinforcing bars. So anyway, so rebar, it's like rebar because this, where you have fibrocartilage, it's where the cartilage is under a lot of stress. It has compressive forces on it, okay? And look at the example. This is pretty good. They have the intervertebral discs here. Those are little cartilaginous discs that are between consecutive articulating vertebrae. And then you think about that in your spine, there's that's a lot of a lot of force pressing on those little bitty cartilaginous disc in your back. And so they're reinforced with collagen fibers to help it stay together. Now you probably heard of herniated disc, or Papa slipped a disc in his back, this kind of talk. Well, these discs, aren't crush proof. They can crush or herniate or whatever. But for most of us, with the collagen fibers in the matrix, and that it holds them together just fine. Okay. So that's why they're there. And that's, the only places that you're really going to find collagen fibers in the matrix of the cartilage is where it needs to be reinforced with this extra strength, kind of holding it together like rebar. So the intervertebral disc, and also in the meniscus of in your knee joint, you think about your legs, that's a lot of weight coming down on those two joints, your knees. And so you've got fibrocartilage in your knee joints protecting the top part of your femur right there, because that's a lot of pressure right there as well. And then also where the two pubis bones articulate with each other in the front, there's a little strip of cartilage right there. The joint itself is called the pubic symphysis. And so in the pubic symphysis, you have fibrocartilage. So those are three main, let me see if they mentioned anything. I can't think of anything else. There's locations: pubic symphysis, intervertebral disc, menisci, points in. Well, that's pretty much it. Yep, that's the only thing I would say. And that's it. Those are your cartilages. All right, now, then it's its own category. They're all connective tissues. They all have chondrocytes, lacunae, and, the only difference is, the fibers, okay? I'm trying to advance here. Oh, come on, you. There it goes. So, you have the connective tissue proper, then the cartilages, and then remember, we have bone in its own category. And bone, remember I said I like to call bone osseous connective tissue because bone, B-O-N-E, is an organ, but it also could be a tissue. And we'll hammer out those differences when we talk about the skeletal system and bone as organs on the next test. But here as a connective tissue, if you look at it, it's quite reminiscent of cartilage, but it's different. You would think that it has to be some kind of relationship for sure, particularly if we just finished talking about hyaline cartilage. And say, where do you find hyaline cartilage? The first place that they list is the fetal skeleton, right? So there's a relationship between bone and cartilage there, all right? And it is true that most of your fetal skeleton that eventually, turns to bone, it's basically just becomes ossified or mineralized in turning to bone. And so there's going to be some very similar characteristics. First of all, right off the bat, you have lacunae. So the cells are inside those little interstitial fluid-filled spaces. You have that. But you don't have chondrocytes in lacunae. In bone or osseous connective tissue, you have osteocytes in lacunae. Okay, so that's a difference, but it's still kind of the same. But one surefire way to distinguish the osseous connective tissue lacunae versus the cartilaginous lacunae is that only in the matrix of bone are you going to have caniculi. Or you remember lacunae means little lakes. Canicula, guess what caniculi means? That's plural for caniculus. And a caniculus is a little canal. So you have little lakes in cartilage, and in bone you have little lakes and little canals. And so why do you have little lakes and canals in bone? Well, the matrix of bone is different than cartilage because it is mineralized. And that's what gives bone its characteristics. It's very hard and rigid. And of course, you want it to be hard and rigid so it can support your skeleton. Okay, so you get a nice, firm, rigid skeleton. And the matrix makes it pretty much impermeable for material to diffuse through it. It's like stone, you know, if you want to build a swimming pool, you know, you could hold water, you could put a cement pool, right? The water's not going to go through there, or some kind of other material, but concrete, cement, stone, whatever, I mean, you can make something waterproof like that. And so, but that's a problem inside this tissue, this osseous connective tissue is a living tissue, and there's living cells in there. And so they have to have a connection to the outside world. They're trapped in their little lacuna, but you get these little radiating channels that come out from each of the lacunae in the form of caniculi. And so that's unique. Now we'll talk about, when we talk about bone as organs, we'll talk about some more detail about what's going on here for these patterns and holes and swirls and circles and stuff. We'll talk more about that later. But for right now, as a connective tissue, osseous connective tissue, a mineralized matrix with osteocytes in lacunae accompanied by caniculi. That's all I want to talk about now as far as that tissue. And of course, representative locations, there's one word, skeleton. You know, that's kind of correct, kind of lame, you know, but it's true. That I'm not saying that's not correct. It is true, because your skeleton, the skeletal system, right? There's different ways you can define the skeleton, right? And the functions of the skeleton and so on and so forth. Your skeletal system, how we're going to define it in my class, is a collection of 206 different organs. You know, you call the bones, right? Those are the organs. But you can have bony tissue in your body that's not really associated with this, quote, skeleton. You know, this is a connective tissue. Osseous connective tissue is a connective tissue. Now, it contributes to the organs, the bones, right? And so most of the organ bone, like your femur, you know, is osseous connective tissue. But technically, there's other tissues in there besides this stuff. So that's what makes your skeletal elements, the bones, the 206 organs. But there, again, there are certain bones, I guess you could say, that or presence of this tissue that's not a part of your skeleton. Like not so much in human, well, there are certain bones that you don't include. And when we study the skeleton, we'll see this, that there are certain bones you don't include, like the 206. You actually have, you counted up every piece of osseous connective tissue in your body, you'd have way more than 206, okay? Because this tissue, if you think about its characteristics, hard and rigid, it's invested in areas that need to be fortified, okay? And we'll talk about what all it is. But for right now, we just say, hey, it forms your bones in the skeletal system, and its function, you know, hey, makes your skeleton, all right? Now, when we talk about osseous connective tissue in the organs and the skeletal system, we'll get into more detail, but that's good for now as far as this kind of tissue, all right? Okay. Now, this is our last connective tissue, which again is a weird one. As I you know why it's in its own category. You commonly refer to it as blood, but in histology class, I don't call it blood. I call it vascular connective tissue because it's a connective tissue. All right, vascular connective tissue because it flows. All right, but you think, how is it a connective tissue? Well, remember, it has all the characteristics of a connective tissue. First of all, it's derived from mesenchyme, and it has cells, and it has proteins, and it has a matrix. Like all the connective tissues, all the bone, all the cartilages, all the connective tissue proper, all have the same basic ingredients, okay? And so what's so special about blood? I think probably the main thing, I guess the biggest difference is its matrix, because it's liquid. This is the only kind of connective tissue you can pour, right? But let's talk about the components. So here, the components of vascular connective tissue or blood. First of all, the cells. All tissues have cells, right? And you just refer to the cells collectively, blood cells, but there's different types, all right? Everybody knows this. Blood is red, right? You look at blood red, red blood, red rum. Remember that movie? Anyway, ask me about that movie sometime. I'll tell you that I actually was going to audition for that little part for Danny in The Shining. And I was going to be the little guy going, red rum, red rum. That was going to be me. But they told me I was too old. I was, at the time, they wanted like a 6 or 7 year old. And I think I was like 11 or something. And they had auditions in Chicago. And I lived just south of Chicago back in the day. And I was going to audition for that part. I could have been a movie star, but no, I was destined to be an anatomy professor. But anyway, here I am. So anyway, red rum. Erythrocytes, that's blood, the red blood cells. Remember we talked about that? And I know these are 3 slides kind of focused on some other cells, but you obviously see red cells in the background. And these are by far the most numerous cell type in blood in humans, and this is true for all mammals. The red blood cells, or erythrocytes, are anucleated, which means they don't have a nucleus when they're mature. When they're first formed, of course, they have a nucleus, but when they're functional cells in circulation, there's no nucleus. You see, look right here. It looks like a red donut, doesn't it? See all these red cells? There's no nucleus. It just kind of looks like cytoplasm, whatever. They're often described as a biconcave disc. So they're not really round like a spherical cell. They're kind of compressed like a frisbee, but really thin in the middle. And so it's so thin that light passes through it. So that's why it kind of looks like a biconcave disc. Well, that's where the nucleus used to be. So they function, they live for a month or 2 in circulation like that until they're recycled, because they can't really do anything without a nucleus other than transport respiratory gases. That's why they're there. Oxygen and carbon dioxide are traveling on the erythrocytes. So that's why you have them, okay? So those are those guys. And of course, the function of all the blood cells, that's a big component of physiology, and you'll get into it more there. I don't talk about too much of that in here. Basically just describe the cells in a histological context, okay? All the other cells, the red blood cells are white blood cells. White blood cells are collectively known as leukocytes. And I think we talked about this before, that leuko doesn't really mean white cell. It's like erythro means red cell, site, get it? Leuko means like yellow cell. And why is it yellow? Well, and they call them white. I don't know. Anyway, but there's other leukocytes. There's about a half a dozen or so different types of white blood cells, but they fall into 2 categories. These 3 across the bottom are collectively called granulocytes, or granular, grainy cells. So if you look in the cytoplasm of all three, it looks kind of grainy. Kind of not real grainy, but a little bit more grainy and really grainy. You see that? And those are the three types. Neutrophils, eosinophils, and basophils are the three types of granulocytes. And the names, neutrophil, eosinophil, basophil, phil means to love or like something. And what are they liking? Well, they're liking the stain. Neutral mean like neutral, does not. They're pretty neutral. Can take it or leave it, kind of like. Well, neutrophil, it doesn't like the stain, so it doesn't really pick up the stain. Eosinophil, it just means like a little bit. Basophil, a little bit more or a lot more. So that's basically what's going on. Now, that doesn't really describe what they do or anything like that. It's just kind of like the appearance under the microscope. So those are your three. But basically, neutrophils and eosinophils are functional in your body's immune system and defense and that sort of thing. Although basophils, here I have in parentheses, the real dark ones, I have in parentheses, those are your mast cells, okay? Or they become the mast cells. What are mast cells? Well, mast cells are the cells in your body that release histamines. So what do histamines do? Well, histamines are responsible for the inflammation response. Like if you get stung by a bee or something and it swells, you get some swelling going on, well, that's your body releasing histamines from mast cells. So you get some swollen, irritated tissue in certain areas, blame your basophils for that. So that's what they are. Of course, you want to counteract the release of the histamines from mast cells. You take antihistamines, right? Does that make sense? Anyway, but these are all your granulocytes, which are one form of leukocytes or white blood cells. Everything else is agranulocyte or without. See, no agranulocytes. It doesn't have these little grains in the cytoplasm. And there's really only two main types, I should say, two types of granulocytes. Lymphocytes, like lymph nodes associated with the lymphatic circulation. Lymphocytes, there's two forms of lymphocytes. They'll either come from the thymus gland or bone marrow. And so sometimes they're further differentiated from where they come from. And that's what constitutes T cells or B cells, or T lymphocytes and B lymphocytes. But they look pretty much the same. So that's a lymphocyte in circulation. And these are real common in the circulatory system as far as white blood cells. But you can distinguish them from any of the other cells in the sense that they have a huge nucleus. You see this nucleus? That's this big dark center. That's its nucleus. It takes up almost all the cytoplasm, okay? No granules in the cytoplasm or anything, but there they are. Big old fat lymphocytes, okay? And then the second type of agranulocyte are called monocytes. And that's the only other type of agranulocyte you have. They're either gonna be a lymphocyte or a monocyte. Now, monocytes typically aren't in circulation that much, although you could see them. But once they're formed, they become macrophages via diapedesis. And what does that mean in English? Well, a macrophage, this is a microscopy talk for you, or like microbiology talk. Macro means big. Phage means to eat. So a macrophage in microbiology is a large eating cell. It's a cell that kind of creeps around and it just eats everything, Well, it eats things that it's supposed to eat if it's behaving correctly. But that's what it's doing. It's part of your body's defense. It creeps around and eats like foreign invaders, bacteria, fungus, dead and dying cells, whatever. It creeps along. Now, diapedesis You see that word diapedesis? That refers to leaving the circulatory system. So the monocytes, once they're formed, they're circulating, but they leave the circulatory system and they set up shop inside tissues. And they're all pretty much the same, just these macrophages creeping around. And wherever they are, they'll have a special name. And we'll name them. I don't want to just give you this comprehensive list. That'd be pretty dry and boring, but we'll talk about certain macrophages when we get to them and the different body systems. All the body systems have macrophages, but those macrophages used to be monocytes, which, remember, monocytes are a type of agranulocyte, which is 1/2 of the leukocytes. Does that make sense? Getting all your ducks in a row? Okay, so erythrocytes, leukocytes, and then thrombocytes. And if you look at the word thrombocyte, thrombo for thrombus, a thrombus is a blood clot. So if you look at thrombocyte, it's a blood clotting cell. That's literally what it means, but there is no such thing. You see what? Well, thrombocytes, they're more appropriately referred to as platelets, okay? And if you look at platelets really aren't cells. Platelets are actually fragments of cells, okay? If you look at this little illustration here, you see these little bitty, you see some red blood cells and everything else is a white blood cell, but then these little bitty pieces of red stuff, those are platelets. Or you can call them thrombocytes, but I think most people call them platelets. And platelets are fragments of cells that are functional in blood clot formation. So they're really not, maybe they used to be thought of as cells, but they're really not. They're just pieces. The cell that they come from is this huge monster right here called a megakaryocyte. So, macro means big, but mega means, wow, real big. This is a megakaryocyte. Megakaryocyte, of course, is a cell that forms from the red bone marrow, and it's destined just to fragment and exist as fragments or platelets in circulation. And of course, it's instrumental in blood clot formation. And that's a whole series of events that takes place to forming blood clots. And again, that's another topic for physiology and beyond the scope of this course. But now you know where it comes from. And so I guess 3 main types of cells: erythrocytes, leukocytes, and thrombocytes or platelets. So that's if you're thinking about this in terms of a connective tissue, those are your cellular components of this type of connective tissue. So what about the matrix? Well, again, remember the matrix is liquid, all right? And so your matrix has a name. It's just called blood serum, okay? Your blood serum is the matrix. It's the fluid portion of the blood. It's mostly water. It's 90% water, 2% nutrients, electrolytes, which are ions, Anytime you got a metal or a salt that dissolves or disassociates in water, it's a charged particle, and a charged particle is an ion. And that's more phys talk. You talk about anions and cations. Those are negative and positively charged ions, or just electrolytes, same thing. Anyway, all these things are part of the serum, all right? And the proteinaceous portion of your blood, the proteins, those are your plasma proteins. So you got blood cells, blood serum, blood proteins, okay, or plasma proteins, all right? What are the plasma proteins? Now, they're not fibrous. You don't have like slinkies in your blood, no toe straps, anything like that. But you do have proteins. The plasma proteins, those, they're important in regulating osmolarity and transporting hormones. Constitute about 8% of the plasma volume. Okay, so they're functional in transporting stuff, all right? Here, and I kind of tell you what blood plasma really is, because sometimes people ask you, what's the difference between the plasma and the serum? Okay, remember the serum is the matrix. The plasma is in the serum. Okay, so here I have the plasma equals the blood serum plus the proteins. Okay, so that's one of the things. Like sometimes the components of blood, you know, when you donate blood, you ever done that? Donate blood? They don't use what they call whole blood for recipients. Usually don't receive whole blood. Like when you need blood for surgery, you normally just get packed red cells. And so they process that blood. The blood that you donate is typically processed in the sense it's separated into components. Sometimes they separate it into just packed red cells and then just the blood plasma. And sometimes they'll separate out the plasma and just have the serum. And so they use different parts of it, okay? So, or if you look at blood, it'll separate. If you look at like a vial of blood and leave it in a test tube, a heparinized tube that prevents it from clotting, it'll eventually will just kind of separate, and you can see different layers of it. That's usually when they're processing, what they do is they'll centrifuge the blood, they'll spin it down, and that's how you get your packed red cells separated from your blood plasma. And if it's heparinized, it'll separate out the proteins, and so you get the serum and so on and so forth. So that's all your components of blood. Now I included this. Did y'all see this right here? Never let monkeys eat bananas. I thought that was kind of fun. And there's always some kind of like little mnemonic, these little rhymes or jingles to help you memorize stuff. And this mnemonic, never let monkeys eat bananas, is in order of most abundant to least abundant white blood cells in circulation. So never let monkeys eat bananas stands for neutrophils, lymphocytes, monocytes, eosinophils, and basophils. These first two, neutrophils and lymphocytes, look at that. N for never. About 65% of all white blood cells in circulation are neutrophils. Okay, you see this one right here? Remember, that's a granulocyte, but with a kind of neutral staining, so not a lot of granules in it. If you look at it, this is, probably most important for students in lab, but I'm just presenting it here anyway. You look at the nucleus, or you probably didn't recognize this. See this squiggly, swirly looking thing in the cytoplasm? That's its nucleus. All granulocytes have a weird lobed kind of nucleus like this, but the neutrophil, it's lobed like that, but there's no kind of granules in it. But like I said, about 65% of all white blood cells will be that. And then the second most abundant are the L for lymphocytes, about 30%. So if you think about the 65% and 30%, that's 95% of all white blood cells are either going to be neutrophils or lymphocytes. And that's true. When I tell students in lab, if you're looking at a blood smear and you're looking at, you can see red blood cells easy because that's almost everything's a red blood cell. Every once in a while you'll see a white blood cell, which would be this kind of large, kind of purplish, bluish kind of staining cell. And most of the time, it almost always, it's going to be either a neutrophil or a lymphocyte. As a matter of fact, I tell students, don't even think about finding anything else because it'd be just a weird, rare occasion if you found anything else. But you can easily distinguish a neutrophil from a lymphocyte by, the neutrophil has that weird kind of lobe-looking nucleus. And then the lymphocyte, remember, that's the one with the large, huge nucleus. If you have a big nucleus, it takes up almost all the cytoplasm. That's going to be a lymphocyte. So here's like a large lymphocyte, small lymphocyte, big old fat nucleus. So that's 95% of all white blood cells. The remaining 5% is divvied up between monocytes, eosinophils, and basophils. So like less than 3% or so are monocytes, and the eosinophils about 2%. Basophils are the rarest. Less than 1% of white blood cells would be a basophil. Okay. So remember, those are just kind of rare ones that you will probably never see. And so, like in my lab, we don't even look for them. So there you go. That's all your connective tissue. Next is muscle tissue. Now, it took some time getting through epithelial tissue and connective tissue because they're very diverse. There's all different types. Muscle and nervous tissue go pretty quick, and we'll be done. And actually, we'll be able to start the skin today, which is good. Because muscle, it's not real diverse. There's only 3 different types of muscle tissue. In nervous tissue, there's only really one type of nervous tissue, and then we're done. But let's talk about it. What makes muscle tissue muscle tissue? So I start off with characteristics of muscle tissue. Well, first of all, it's highly cellular, it's vascularized and innervated. So right there, that's kind of like a combination of epithelial tissue and connective tissue. Remember, epithelial tissue was highly cellular, but it was never innervated or vascularized. But muscle tissue is. Lots of cells, lots of blood vessels, lots of nerves, okay? But this is the second characteristic is probably hands down, or at least in this class, how I teach it in this class. Muscle tissue is the only tissue that can do this. It's the only tissue capable of responding to a stimulus by contracting. So it moves. And so all movement of your body involves some kind of muscle. Okay, so consequently, muscle contraction is responsible for all movement of the body. And so there you go. It's involving some kind of muscle. And then so, that's, if you want something to move or whatever, you have to involve some kind of muscle. And there's 3 different types of muscle. Different parts of the body do different things, okay? There's skeletal muscle, cardiac muscle, and smooth muscle. Now, you look at them, they look pretty different. They're all, and we'll look at these in lab. Skeletal muscle, that's, you think skeletal, like skeleton. These are the ones associated with your skeleton. So this is your voluntary muscle that you have conscious control over. If you're like moving on purpose, talking, blinking, jumping up and down, breathing, whatever, you're using skeletal muscle. And most of the time, it's attached to your bones. Skeletal muscle can be attached to bone, it can be attached to other muscles, and it can be attached to your skin, so whatever. But it's generally, you think of it as the voluntary muscle. The other 2 types are involuntary. You don't have conscious control over them. They're all governed by the autonomic nervous system. But only the skeletal muscle is governed by the somatic motor system, or the voluntary nervous system. Now, if you look at it, it's unique compared to the other 2. This is a lateral view looking at 1, 2, 3, 4 muscle fibers. When you're talking about muscle in the terms of histology, the tissue, the word fiber is synonymous with the word cell, for muscles, that is. So muscle fibers are actually muscle cells. They call them fibers because they're huge cells. So this is a cell, this is a cell, this is a cell, this is 4 muscle cells, 4 muscle fibers. And you'll look at all those dark spots. What are those? Those are nuclei. And so they got the whole bunch of nuclei. See that? So there's 4 cells or 4 fibers, but a whole bunch of nuclei. So skeletal muscle is multinucleated, which means there's more than 1 nucleus per cell. It's the only type of muscle like that. None of the other muscles are multinucleated, only skeletal muscle. And another thing too, if you look at it from this view, you see, if you look carefully, you can see these vertical little stripes. or striations. You see that? That's not unique to skeletal muscle, but it is very apparent. And so we often describe as the fibers of skeletal muscle as being striated. So a striated muscle has these stripes on there. What is that? Well, that's an artifact of the contractile proteins that are in the cytoplasm. Just the arrangement of them, how they're oriented in there, because it's all these proteins. And you'll learn about the function of muscle contraction, all that in phys again. But we don't talk about that in here. But that's what you're looking at. Those alternating dark and light bands are the proteins, and that's it's just an artifact of the skeletal muscle. And so you can see that. So multinucleated, striated, voluntary muscle, skeletal muscle. Bam, there you go. Of course, where you find it? Well, you find it in your skeletal muscles. You have the muscular system. It's comprised of about 656 named muscles. There you go. So any of those are made out of this stuff, all right? Now, technically, your skeletal muscles are organs. So there's other tissues involved, but this is the main tissue, just kind of like bones, like bones. There's osseous connective tissue, but there's other tissues involved that makes them organs. All right, so there you go. Functions, locations, I think I covered that. Function is, voluntary movement or whatever of your muscles, locations in your skeletal muscles. Now, cardiac muscle, take a guess. Cardiac muscle, where do you find that? In the heart. That is true. You find it in the heart, but also a little bit of it in the large arteries that are coming out of the heart. When we talk about the circulatory system, we talk about these arteries. There's two main ones coming out of the heart, the aorta, obviously, and the pulmonary trunk. The aorta is carrying the oxygenated blood into your body, and the pulmonary trunk is carrying the deoxygenated blood to your lungs. But they're arteries, all right? And so they're continuous with the real meaty muscular pumping chambers of the heart, the ventricles. And so the walls of the ventricles are cardiac muscle, and then the walls of these arteries have a little bit of cardiac muscle in them as well. So what does it look like? Of course, we know what it does. It contracts, and so it propels blood, circulate blood. But if you look at it, this view is just kind of weird looking. So that's why it's nice to have an artist rendition or a little diagram to see what's kind of going on. It's not, the cells are not multinucleated. They're typically uninucleated, which means one nucleus per cell. The cells are often described as being branched. If you look carefully, it kind of looks like a pair of pants coming and splits. You see that? Splits, and it connects to another one that splits, and they kind of split. Those are the branching cells. Branching, uninucleated cells, and they are striated. It's striated kind of like skeletal muscle, but not quite to that extent. So it's, I would say not heavily striated. I would say skeletal muscle is heavily striated. Cardiac muscle, lightly striated. But there is one thing that's hands down unique about cardiac muscle under the microscope that you would see that you never see in any other type of muscles, and that's the presence of these vertical lines here. You see that dark line here, dark line there? Those are called intercalated discs. Only cardiac muscle has intercalated discs. What is that? Well, where one cardiac muscle attaches to another cardiac muscle, it forms this intercalated disc. It's like a linkage. The analogy I always give here is it's like linkages on boxcars. You got like a choo-choo train. One boxcar connects to another boxcar, and that boxcar connects to another boxcar, and so on and so forth. You have one engine in the front, and when you pull, the engine pulls, it pulls this car, and this car pulls that car, and this car pulls that car, and whatever. So that's kind of what the intercalated discs do. The intercalated discs ensure when any one of the cells are stimulated to contract, all of them contract at the same time. And so you want that in the heart because you want the heart contracting in unison. And so that way it's more optimal contraction of pumping the blood. And on, on paper, I guess you can say, it works beautifully, but sometimes it doesn't contract in sync. And when it's contracting out of sync, they have a name for that. It's called fibrillation. You're talking about infib, you got atrial fibrillation and ventricular fibrillation. It's not a good thing. Atrial fibrillation is not quite as serious as ventricular fibrillation, but if your ventricles are in fibrillation, you're going to die. It's deadly. And so if so-and-so is in fib, what do they got to do? How do they correct that? They defibrillate it. How do you defibrillate? Well, with a defibrillator. You know what a defibrillator is? Those things, they watch those hospital shows, and they put those paddles together, they put them on chest, like clear, and they boom, and they shock you. That's the defibrillator. Well, what does that do? Well, it sends electricity through the body, high voltage, low amperage. And basically it kind of resets the electrical currents in the heart. So the pacemaker kind of send a new current through. You'll learn all about that in phys, and it's interesting stuff, but we got to talk more about histology. Okay, so cardiac muscle, branching cells, uninucleated, mildly striated, presence of intercalated disc in the heart, in the large arteries, tracks blood, blah, blah, blah. I think you got that? That's pretty good. And then finally, the last type, smooth muscle. Smooth, it's smooth. Why is it so smooth? You know why it's so smooth? No striations. It's no non-striated or unstriated, not unstriated, just non-striated, uninucleated cells. That's what these are. There's one long kind of tapering nucleus per one long kind of tapering cell. The cells they form broad, thin sheets of muscle mostly in the walls of hollow organs. And that's why smooth muscle is sometimes called visceral muscle. Visceral muscle means of viscera, and viscera, your organs. And so in the walls of your hollow organs, you're going to have muscle, visceral muscle or smooth muscle. And where, skeletal muscle was voluntary, smooth and cardiac are involuntary. You don't have conscious control over how your heart beats or how your muscles contract or whatever. You don't. So it's governed by the autonomic nervous system. But smooth muscle, you can distinguish it from the other two. Well, it's uninucleated, but it's non-striated. That's pretty unique. Long tapering cells, broad thin sheets in the walls of your hollow organs. I guess you could say your heart's a hollow organ, but it's got cardiac muscle in it. But this stuff, it's in all organs, really. If you have a hollow organ, stomach, small intestine, large intestine, esophagus, trachea, urethra, vagina, rectum, whatever, hollow organ. Histologically, you look in the walls, you're going to find smooth muscle, because you think about the hollow organ is hollow for a reason. Something is going through that hollow space, and it doesn't just go through there based on gravity. Something is pushing it or propelling it through there, and it's going to be smooth muscle. Okay, so the involuntary movement of material through some kind of passage or whatever. So think of an organ, it's going to be there. I think that's it. That's your muscle. Now, nervous tissue. This is our fourth and final type of tissue. Again, you give you some characteristics of nervous tissue. It's highly cellular, little extracellular space, so that's not real descriptive or whatever. But there's only really two types of cells. This is probably the most important thing. The numero uno cell, or they're collectively referred to as neurons, or you say a nerve cell. Nerve cell, neuron, same thing. Nerve cell, the functional cell. The neurons are the excitable nerve cells, which actually transmit the electrochemical signals, or your nervous impulses. Okay, so they manifest and send nervous impulses. Okay, that's it. Your neurons. Everything else that's not a neuron in the nervous system, all right, or in nerve tissue, would be a neuroglial cell. Sometimes they're just called glial cells, which is kind of short for neuroglial, but the word neuroglial literally means nerve glue. Nerve glue. We think about that. What's glue for? What do you use glue for? I need some glue. Why? Well, I'm gonna put something back together, glue something, so hold stuff together, right? Now when you study the nervous system, particularly the physiology of the nervous system, there's going to be all kinds of other cells that are supporting cells. They're in the nervous system, but they have nothing to do with manifesting and transmitting nervous impulses, all right? So if they're a excitable cell, they're a neuroglial cell. So here I just have smaller supporting cells, non-excitable. Here on this, this is a nerve smear, and we look at this in lab. You see this big blob-looking thing with all the little branches coming off of it right there. This is one neuron, okay? That's your nerve cell. And you can see there's this little nucleus. All these little branches are processes of it. There's two types of processes that come off the neuron. The highly branching dendrite. The word dendrite means little branch. Bunch of these little branches. This is a sensory portion of the neuron. So if information is coming into the cell, it comes in through the dendrites. And then usually opposite that, you have a large extension that comes off that's actually going to transmit a nervous impulse, and that would be the axon. So all neurons have axons and dendrites attached to them. Dendrites are receiving information, dendrites are transmitting information. And basically the center part of the cell, where you have the cytoplasm and the nucleus and everything like that, that's at the body of the neuron, or the soma. The word soma means body. Sometimes they call it the biosynthetic center of the cell, but just the main part of the cell where you'd have this cytoplasm and all the other organelles and so forth. But the unique thing is that you got axons and dendrites. The other nerve cells, the neuroglial cells, don't have anything like that. They're supporting, there's different types. We'll talk about different types when we get to them. But that's basically it. On a nerve smear slide, how they prepare a nerve smear is basically, for most of your labs, they take the spinal cord from a cow and they blend it up. They liquefy it and then put a drop of that liquid on a slide and make a smear. And so this is what you see. All those cells kind of streaked across the slide. This is kind of, the neurons, they'll look different. You know, they don't look like this all the time. They're just kind of a random, just like you look at them under a microscope, you'll see. But they'll have axons and dendrites. And the little bitty specks you see kind of scattered around, those are all the little neuroglial cells. It says nuclei of glial cells right there. And that's basically it. The neurons and the neuroglial cells form all the structures of this CNS and PNS, which is the central nervous system and peripheral nervous system. So your brain, your spinal cord, all your nerves made out of nervous tissue, which are neurons and neuroglial cells. The end. So that's it. That concludes all of our histology. We got anatomical terminology, histology, and one more thing before your first exam, and that's the integumentary system, which, you know, we've got to complete that before your first test. And your first test on paper, you know, I always say I'm going to have it on the 4th week class. I never make that happen. It's seldom. It's going to be, so, well, we can do it next week, but I know you have your practicals next week, right? Right? You want to have a practical and your electric exam on the first, at the same week? You do? you're not going to be very popular. Got one student that wants to take it all at the same time. Just get it over with. Well, we'll see. But what I have to do is finish the skin first, and will I finish it this week? I probably could during today, because we have we got about 10 minutes. Is that right? I can put a dent in it in 10 minutes, but I won't finish it in 10 minutes. But between today and Thursday of this week, I can probably finish. So maybe next week, but we'll see. Y'all think about it. Thursday of next week, that might be not Thursday or Tuesday. What would be better for you? I don't know. Let's talk about this later.
Practical is next Friday.
So what?
Our test for lab is next Friday.
I don't know. Ask me about lab stuff in lab.
Okay.
Because I don't know. But yeah, all laboratories. I don't know when you take labs. That's why I'm saying that. But all labs have my sections, everybody else's sections. They should have all laboratories are being tested next week. No, next week. Yeah, next week. Yeah, so all my sections for sure. If you go on my courses and look at your lab practicals are up there, it has next week's date on there already. So remember, it's gonna be online, it's on there. So yeah, it's on there. It's already scheduled. But your lecture test is not scheduled because I don't know. I gotta finish. Find it first. Your skin, the integumentary system. Let's see. I'm looking in the wrong place. Let's see. There we go. I'm gonna open it up. Integumentary system. Well, I love this. It's like every thing I talk about is my favorite thing while I'm talking about it. But I really do like the integumentary system. That was I'm sure I told you guys this before that I'm a zoologist. That's my background. I'm a vertebrate zoologist, and so that's somebody who studies animals, right? But a vertebrate zoologist studies animals with backbones. So you studied everything? Well, I've done work with just about everything, but my main animals of study were frogs. And of frogs, I've done a lot of work with toads. And specifically, the behavioral thermal physiology of toads, like how the toads control their body temperature by altering their behavior. Remember, like toads and frogs and other things are cold-blooded, but we say that. You say things that are poikilothermic, they're not homeothermic, they're poikilothermic. They can't really control their body temperature. Well, that's not 100% true. And so that's some of my research. My thesis research was studying how they can actually influence their behavior or influence their body temperature, influence their behavior. And there's a lot of association between an animal's body temperature and their integument, including mammals. And so I've done a lot of work studying the integument. And so I don't know, I kind of like the integumentary system of mammals and then frogs and toads and all kinds of things. And so I don't know, this is kind of a fun thing for me. I like talking about the skin. So without further ado, let me talk about the skin or the integumentary system. I got like 6 minutes to get started. So let me do that. Let me fix my display here. And so here we go. Integumentary system. This is our first body system. Are you excited? Our very first body system. We're talking about all body systems this semester. Integumentary comes from the word integument. So what is an integument? An integument, just a regular integument, is some kind of covering. You have a covering of something, and all living things. It doesn't matter if you're one cell big or multicellular or live in the ocean or live on the land or live wherever. All living things have an integument of some sort, some kind of covering that separates the internal contents of their being from the outside world. And that's what we have. We have a system in our body that does that. It interfaces with the outside world. And so that's our integumentary system. And so what are the components in the integumentary system? I like to think of the integumentary system. You say, oh, just your skin. Your skin. There's more to it than just your skin. You may have heard people say that your skin is the largest organ in your body. Yeah, I guess you could say that. You know, pound for pound or unit per square area. You know, there's more surface area of skin than anything else, and big, whatever. I don't know. Is that true? Probably. But it's big. It's one big organ, I guess. As far as the anatomy goes, the system, all right, there's two main parts of the integumentary system. First, the skin. The skin is actually the membrane, the cutaneous membrane. A cutaneum or cutaneous just means skin. The words are interchangeable. Your cutaneous membrane and cutaneum, that's your skin. And so of the integumentary system, that's the main part is the cutaneum or your cutaneous membrane. And everything else that's not really a part of the skin per se would be accessory structures of the skin. Okay, like what? Well, all your glands, your integumentary glands, your cutaneous glands, all right? Your hair is considered an accessory structure of the skin. And your nails. So it's the main thing, three main things. Your nails, your hair, and your glands are accessory structures of the skin or part of the integumentary system. And we'll talk about all these, all right? So let's start with the regions of the skin of your integument membrane. There's two regions of the skin proper. There's that word proper again. Remember I talked about word proper, say in the strict sense, how we define the skin. So really the skin, the integument, the cutaneous membrane, there's only really two true parts. And we've talked about these in passing when we were talking about histology. There's an epidermis and dermis, period, that's it. And then everything else that's not epidermis or dermis would be regions deep to the skin. That would be the subcutaneous layer, or sometimes the word hypodermis is used. But if you look at the word subcutaneous and hypodermis, you're saying the exact same thing with just kind of different words. They're synonymous. Subcutaneous means literally under, the cutaneum, the skin layer, or hypodermis. Hypo means under or below, and of course dermis, under the skin. All right, so hypodermis, subcutaneous layer. It's part of the integumentary system, okay, but it's really not a part of the skin proper per se. So it's really not a part of the skin as an organ. It's the stuff that's underneath it, subcutaneous, right, or hypodermis. So there's that. So we'll talk about the regions. Let me see. We've got like 2 minutes. Do I want to do this in 2 minutes? Let me see something here. Where am I? Yeah, let's save this Thursday. That'd be a good place to go. So we're going to begin. Is that right? Yeah, that's a good little intro. And then Thursday, we'll do this characteristics of the region. Talk about the epidermis. We'll define each of these regions. And then we may or may not, I don't know, we'll see how far we get. I might be able to finish all this Thursday. Maybe not. Then we'll talk about a test after that. So tune in Thursday. That's going to be an important lecture because we're getting close to our first exam. Okay. All right. Y'all have any questions about anything? No? I guess you're saying no. I'm going to quit there for today and see you guys Thursday. And I'll see some of you guys in the lab. Okay.